All right, so we are on chapter 8, section 6, and we're going to talk about osmosis and diffusion. So if you took some introductory biology, this should sound pretty familiar. So osmosis is movement of water. So we know that our bodies are mostly water, right? And where is that water? Well, that water is partially inside the cell and partially outside the cell. And so we call the solutions that have water, we call them aqueous solutions. That's why we wrote AQ behind all of our equations. So in these aqueous solutions, we have different things dissolved inside of them, like sugars and ions and all kinds of things like that. And so the concentrations of the solutes in these solutions is very, very tightly controlled. And so they're usually different between what's outside the cell and what's inside the cell. And so the way that these two aqueous solutions are kept separate are by our cell membrane. And our cell membrane is what we call semi-permeable. So some things can get across just by diffusion and some cannot. And so the things that are barriers, right, is that phospholipid bilayer. Right? So some things can cross the phospholipid bilayer without any help, and some things need help. And so under physiological normal conditions, we say that these solutions are isotonic. So if something is isotonic, what's on the outside is equal to what's on the inside. And so a lot of times when we talk about that, we're thinking about water and the things that are dissolved in the water in terms of inside and outside of the cell membrane. And so you have to think about this in terms of what would happen if I change those physiological conditions. So what happens if I drink a whole lot of water? Well, when I drink the water, it's going to go into my digestive system, and then it's going to get absorbed from my digestive system into my bloodstream. So it will actually dilute my blood. So that means that the concentration of solutes in my body is going to go down. So then there's going to be an imbalance in the concentration of stuff or the concentration of solutes that are outside the cell versus the concentration of the solutes inside the cell, right? So you're going to have less outside and more inside. And so in this case, we call this solution we call the solution outside the cell hypotonic. And so what's going to happen if you have a hypotonic solution is that water is going to actually move across the membrane to equalize concentration. So how's that going to work, right? Well, if I have a low concentration here and a high concentration here, what's going to happen? My water is going to pass through the semi-impermeable membrane in a process that we call osmosis. And so water is actually going to enter the cell, and the cell is going to swell and could even burst if it gets to be too big. And so what I like to think about is I have like a beaker full of water, and that's going to be my blood. And then I have a cell. And so this would be a hypotonic solution where we have a low concentration of SOLUTE of solute in the blood, so the blood. And then inside the cell, we have a high concentration of solute, right? So what's going to happen? The water is going to flow into the cell. So water into the cell. Okay, so the way that I remember this, hypotonic has the word pot inside of it. So when I think about what's going to happen if I'm going to go cook and I have a pot, here's my pot, I'm going to put on the stove. What do I have to do in order to cook? I have to put water into my pot. So if water is entering the cell, it's a hypotonic solution because water goes into a pot. All right. So if too much water enters that cell, the cell's a certain pressure, and the more water that enters the cell, the higher the pressure, and eventually you can't put any more water in and it's going to burst, right? So that's called cell lysing. And so you have to think about however much water is going to flow through that semi-permeable membrane, right? It's going to exert a pressure on that membrane. So that pressure that eventually would make the cell lyse, we call that osmotic pressure. So the more concentrated the stuff inside the cell is, the more water is going to come in. So the higher the osmotic pressure. Pure water, right, with no solute inside of it, is going to have an osmotic pressure of 0 because nothing's going to change, right? It's not going to apply any pressure because there's no solute there. So if you apply pressure in opposition to the osmotic pressure, then you're going to stop that cell from lysing or from exploding, right? So when we look at the concentration of dissolved ions in salt water, right, it's about 3 times out of blood. So if we're stranded on a deserted island, right, there's water surrounding us, but you can't drink it. Why can't you drink it? Because if you drink that seawater, and that seawater has 3 times the amount of salt, when we drink it, what's going to happen is that the water is now going to move from our cells into our bloodstream. Well, that's not good, right? Because then the cells themselves are going to lose their water, right? They're going to lose it, and they're going to get smaller, and they're going to get smaller, and they're going to get smaller, and they're going to shrivel up, and they're going to die. And that's crenation. And so this is why most, we're surrounded by water, but most of the water on the is not drinkable water. And so that's a serious problem, right? But here's my same analogy, right? I have my beaker, and inside my beaker, I have my cell. So if you have a high concentration, so if you have a high concentration of solute in the blood, right? And then you have a low concentration of solute in the cell, what's going to happen? Then we know our water will move out of the cell. So this is how I remember, you're going to laugh at this, that if something is, this is hypertonic solution, right? In a hypertonic solution, our water is going to move out of the cell and the cell is going to shrivel. So how do you remember that's hypertonic? Okay, what's in the middle of hypertonic is the word per, right? And so what I think of, I'm going to try to do this. It's going to be so sad. When I think of something purring, I think of a cat, right? There's my little cat's face, and this is terrible, but there's his whiskers, right? And okay, and his body, oh, that's a fat cat. All right, so what does a cat do? A cat purrs, and that's a sound that comes out of the cat, right? So if water is moving out of the cell, that is hypertonic, because a cat will purr out, right? And so that's kind of how you can remember hypertonic and hypotonic. If the solution is a hypotonic, water will enter the cell, like water goes into the pot of water, right? But if it is hypertonic, the water is going to come out of the cell because the cat purrs, and that's a sound that comes out of the cat. All right, so this is what really happens, right? There's no cats and pots and that kind of thing. But if you put a red blood cell in an isotonic solution, so this is like putting red blood cells in plasma, right? It's isotonic. It's the correct concentration of solutes. Nothing's going to happen. It's not going to change the pressure. The water's not going to enter the cell. Water's not going to leave the cell. Nothing's going to happen. That's normal. So that's an isotonic solution. Now, a hypotonic, remember hypotonic, we have our little pot, right? And you're going to put water inside the pot. So the water is going to enter the cell because you have lots of solute inside of the solution, right? And so, so the water is going to go, I'm sorry, I think I said that wrong. You have let me do this, if I write it down. So you have a high concentration of solute in the cell. So the water enters cell, right? To basically dilute the solute. If you look at your hypertonic solution, you have a high concentration of solute in the blood or in the solution, whatever, and you have a low concentration of solute in the cell. So what's going to happen here is our water will leave the cell, just like a cat purring leaves the cat, right? So either way is not good, right? And drinking too much water is usually not a problem. I mean, you would have to drink a lot. I'm not going to give you, I don't know, it's like definitely more than 10 liters of water a day in order to get to the point where your kidneys can't handle it. But that usually doesn't happen in adults. What we really have to worry about that is with infants, right? If their formula is mixed incorrectly, like if they get too dilute of a formula, there's not enough food in it and it's too much water, they can go through hemolysis. So for them, really, hemolysis is something you have to kind of worry about, right? But the crenation is when you would drink salt water. That would be equally bad, right? So how does this work? Well, during osmosis, right, water is going to flow through our cell membrane. There does not have to be something there to transport the water. It happens. There are also pores that are like tunnels that the water can flow through. So it can be either they just cross the membrane by themselves or they go through basically a tunnel. And they're always going to move from an area where the solution is more concentrated to an area of the water is going to move from high to low. So you got to think about concentration of the solutes. So here, this is a low concentration of solute, right? Ooh. And this is a high concentration solute. I don't really like how they just say solution. I would prefer if they would say solute, right? So where's the water going to flow? Well, the water is going to flow this way in order to go and dilute here so that you now would have equal concentration, right? So then the diluting of the solution is now going to equal the solute concentration on either side, right? Okay, so when we have patients, we have to worry. We have to make sure that whatever fluids we put into their body, that they are isotonic to what's going on in the patient. So we have to make sure that those IV solutions have the correct solute concentrations equal to what's inside of their cells. So if you have an isotonic solution in your IV bag, you're going to minimize osmosis. You're not going to have water crossing the membranes, right? So this is important. This is something you need to know, right? You need to know the common IV solutions. So usually you're going to have 0.90% mass by volume salt in ACl. So we just abbreviate that NS. And if you see capital N, capital S, that's what it means. You usually have 5% mass by volume glucose. And so we refer to that as D5W because the sugar is dextrose and there's 5% of it and we dissolve it in water. So it's a D5W. These are our common physiological solutions that make the IV fluid isotonic. So sometimes instead of expressing things in concentration of molarity, when we're talking about physiological solutions, we express them in terms of osmolarity. So it's really the molarity times the number of particles in solution. So the osmolarity takes into account our number of particles that are exerting a pressure against the membrane. And so if you were to look at blood plasma has an osmolarity of 0.3 osmoles per liter. So that's one of the things that you might see. Now, think about what happens with those solute particles. If you have some solute, right, and it's very concentrated, just like our drink mix that we used that analogy before. If we put a drop or two of that highly concentrated drink mix, right, it's very, very, very dark, whatever color. In this case, it's green, dark, dark green. And we put 1 or 2 drops and we just wait. Don't do anything. Don't even mix it. Just wait. Eventually, hold on, y'all. So no matter what happens, you don't have to stir it, nothing. Just through diffusion, those particles of the solute that are green are eventually going to disperse. They're eventually going to reach an equilibrium in that solution. And so what you started out with was like 1 or 2 drops of this really, really dark, dark green, and you put it into a big beaker of water. And so now that big beaker of water is light green because the resulting solute has distributed evenly throughout that mixture. So this is a spontaneous reaction. It happens. You don't have to put any energy in to make it happen. It's just that those solute molecules diffuse into the water, and they're constantly moving. They have particle vibration, so they're constantly moving, and they just mix, right? And so we can take advantage of this fact, and we can treat patients knowing this. So we know that in our bodies, our kidneys are the organ that filters our blood, right? You know that your blood delivers oxygen to all of your cells. But what it also does is it picks up waste particles that your cell produces. Your blood picks that up, takes it to the kidneys. And so inside the kidneys, you have small waste molecules that are diffused from the blood into the kidneys. And this is where the kidneys turn that waste into urine. And so your cells, all your large stuff basically stays inside of the blood, and your waste particles are what goes into the urine. And so why does it move that way? Well, because it's moving from an area of high concentration of waste inside of the blood to an area of low concentration inside of the kidneys. And so that process that occurs inside the kidneys is called dialysis. And you might have heard that word before, but if it's just dialysis, this is your kidneys actually doing it. And so there are some people who have kidney failure, right? Their kidneys don't function. Well, if your kidneys don't function, your kidneys are not going to filter out the waste particles that should be filtered out. They a lot of times don't filter out the excess water. And so you don't survive for very long if you don't do something. And so what you can do is you can perform hemodialysis, which is blood dialysis, to cleanse the blood. So in this process, right, you put an IV in the person's arm, right, and the blood is removed from the patient, and it goes into a machine. And so inside of the machine, it looks very similar. Well, I don't want to say that, but it has a similar structure to the cell, right? It has a semi-permeable membrane. And so on one side is your blood, right? And the other side is an isotonic dialyzing solution. And so the solution is isotonic for healthy, clean blood. And so you have waste particles of blood over here. And so what happens is the waste particles will go out, but the red blood cells and stuff will all stay in. And it's the same concentration of all the other stuff that should be there, like the right concentration of sodium, the right concentration of all that other stuff. So the only things that will move are the things that you want to get rid of, because they're high concentration here and you don't have any over here. And so all the waste particles will diffuse away, and then the blood will be put back into the patient's body. The clean blood will put back into the patient's body. And so this is kind of what it looks like, right? And so you have one tube that's taking blood out, and it goes into the machine, and it goes in through all the different filters and things like that. And once it's clean, it comes back through another tube into another place on the arm. And so what you see is here's the blood, and it's got this yellow particle, and the yellow particle is the waste product. I don't know why it's doing that. So there's the waste product. And then, so it goes through this solution. So it's inside this membrane, right? This tube is a semi-permeable membrane. And there are no waste particles there to start with, but the waste particles will move out of the blood inside of this semi-permeable tubing, and everything else that's supposed to stay in will stay in, and then it will get returned to the patient clean. Okay. Oh yeah, so let's go do some problems real quick. All right, so we only have a few, which is really nice. All right, we have a cucumber. We're going to place it into a briny, another word for briny is saltwater solution, to make a pickle. What's going to happen? If you pickle something, I want you to think about it before we start answering it with science. If you pickle something, what's going to happen? It kind of gets shriveled, right? Okay, so keep that in mind. All right, does water leave or enter the cucumber cells? Well, think about it, right? If you put your cucumber into, we're going to call it a beaker, and instead of a cucumber, I'm going to call it a cell, right? Because it's the same thing. So what's going to happen is this liquid here is high salt. My cucumber is low salt, okay? So what's going to happen with the water? How's the water going to move? Well, my water is going to move to my area of higher solute concentration. So it's going to move from my low salt to my high salt. So this is the direction of water movement. So water is going to move out of the cell into the solution. So what do we call that when water moves out? So all this water is moving out. Remember our little kitty? I don't know why I like to draw. I always seem to draw fat cats. I did have a fat cat though. And there's his tail, right? Okay, maybe he's just scrunched up and that's why he looks chubby. All right, so this is, if water is moving out, what comes out of a cat is a purr. So this is hypertonic. Okay, so now let's read our question. Does water leave or enter the cucumber cell? Okay, well, we just said it leaves. Does the cucumber swell or crenate? So this is the same thing as shrivel. So what does it do? Does it swell or shrivel? If water moves out, it's going to shrivel. And is the saltwater solution hypertonic or hypotonic to the cucumber? We just said it's hypertonic. Okay, so if you just kind of do these with that little, draw it out, answer your questions before you kind of read them, it'll help you out. All right, we have a patient undergoing hemodialysis. As the blood leaves the patient, its solute concentrations are, well, they've been living, right? So their blood has been picking up waste products. Their solute concentrations are waste. So they are a high concentration. So there's a high concentration. If you have a high concentration of that solution, then you're going to be hypertonic, right, to the dialyzing solution. As the blood re-enters the patient, okay, it's been cleaned out. We've removed a lot of those solutes. Its solute concentrations are, we want it to be equal to what is in the current body, so then we don't injure the patient further. So it's going to be isotonic to the dialyzing solution, right? Okay, let's see. Are the following solutions isotonic, hypertonic, isotonic, hypotonic, hypertonic with physiological solutions? Remember I said you had to know this? Do you remember what it was? It was 0.90% mass by volume of NaCl, right? And 5% mass by volume D-glucose, right? So this is D5W and this is NS, normal saline, right? Okay, so 10% mass by volume sodium chloride. If I have 10% mass by volume sodium chloride, is that higher or lower than my normal sodium chloride solution? That's quite a bit higher. That's way higher than it should be. So higher is hyper. Hypertonic is lots of stuff inside of it. Okay, a 0.9% glucose. Well, if I go look at my glucose is supposed to be 5%. That's a whole lot lower. So that's hypotonic. 5% mass by volume NaCl. Oh, that's a whole lot more. I only have 0.9 of sodium chloride. So this is going to be hyper. And then 5% mass by volume glucose. Well, that's exactly the same. So this is isotonic, right? Okay, and that's it. All right, have a great rest of y'all day.